Na minha escola, cada aula me leva mais longe. Aprender lá me abre as portas do mundo todo. Do Canadá para onde eu quiser ir. A gente aprende brincando.
A gente aprende investigando. We learn in 2 languages. We learn for life. That's the conclusion of my work. E a gente ama aprender onde quer que a gente esteja.
Na Maple Bear, o aprendizado é para a vida, com ensino multicultural que prepara seu filho para ser bem-sucedido em qualquer lugar, no Brasil e no exterior. Escola bilíngue com imersão em inglês e uma das melhores metodologias do mundo. Maple Bear, escola canadense de verdade.
Hello, everyone. Good evening. It's so good to have you with us today. My name is Rafael Mangini. I am Vice President of the Brazil-Canada Chamber of Commerce, and I would like, on behalf of all the boards, to welcome you all to this event. And of course, on behalf of the Education Commission, who has worked a lot to put this event together. The Tech Journey is a very interesting initiative that started with a very interesting group of people who were really curious about the trends of the future, technologies that are going on, things that are happening, things that will happen in the future, and new technologies that are being developed and which will change our lives in the future. And possibly we are not even aware of things that are coming in the next few years. So the idea of the Tech Journey was to bring to you all the opportunity of hearing from some of the most talented and most renowned teachers from the University of Montreal, working in Canada, to bring in updates and interesting things that are going on in the world. So if you would like to listen to this transmission, to this event in Portuguese, you just can go down in the interpretation bar in Zoom, and we are having simultaneous translation to Portuguese. So please feel free to use it. And without further delay, I would like to call Professor Christian Barron. Professor Barron has a doctorate in microbiology from the University of Munich, and he's the head of biochemistry and molecular medicine at the University of Montreal, and he's going to share some very interesting news with us all. So welcome, Professor Barron. Thank you so much for being a part of our event today. And so the camera is yours.
Thanks very much. Thanks very much for the invitation. I'm honored and very pleased to be invited, and especially I seem to be the first presenter of this Tech Journey. But I can confirm that you found some very interesting speakers from different fields at my institution, and we're very pleased to be here to present our most recent research topics, which are certainly pertinent for all of us. And we will be more than pleased to answer questions after the talk. And today, my talk is going to be on antimicrobial resistance. unfortunately cannot speak Portuguese. I could do this talk in English and French and German. I could maybe do it in Spanish a little bit, but not in Portuguese. But yeah, I'm sure the translation will take care of that, and those who will, for whom I speak too fast in English will certainly be able to follow, and I hope you find it interesting. So, okay, let's get started. So the topic of today's talk is going to be from one pandemic to the next, how to counter the resistance to antimicrobial drugs. That's the topic that was chosen. And we talk a lot about pandemics these days. Pandemics kind of a pandemic is influencing our lives right now, but it's of course it's a very important development, a very important pandemic, and we had lots of lessons to learn from that. That said, there's other health issues, other health concerns which are important now and will be in future, and certainly the resistance to antimicrobial drugs is something which will be on our agenda, on our horizon. It should be on our horizon for the time to come. This first slide here kind of gives you an overview of what I will present this evening. After a brief introduction, I will talk about chicken. Well, chicken, most of us eat chicken, and you may or may not know that chicken and the way we grow chicken, we cultivate chicken, is quite important, quite intrinsically linked to the history of antimicrobial resistance. I will then make a broad tour about the history of antibiotics and the development of resistance. I will then go a bit more into detail, and I understand there's several potential graduate students who might be interested in studying maybe even at the University of Montreal in North America in future. So we'll go a bit deeper into mechanisms without hoping not losing most of you into the biochemistry of this process. And I will finalize by speaking about research opportunities at the University of Montreal. I will mention a couple of colleagues who have really, there's many people I could mention, but I picked a few of them who have particularly interesting research programs, just to give you an example, just to give you a taste of people who work at our institution. So we talk about the next pandemic. So what does this mean? So when you look at this slide here, this reflects that what many people think, that the resistance to antimicrobials, and this is a topic I will explore further, of course, is something like a next pandemic, but it's not a pandemic like the one we are living right now. The viral pandemic, COVID-19, you see the very rapid spikes and falls, and rapid spikes and rapid falls. We have now in the fourth or fifth wave, depending where we live right now. Viral pandemics have a characteristic that they are very fast. Antimicrobial resistance is a process. It's a preoccupying development which has been in the making for many years. It is slowly rising, and constantly rising. So it's something we don't see and perceive as a threat as acutely as a viral pandemic. That said, it's still, and I hope that that's something I will transmit, that this antimicrobial resistance is also a development which is to be taken very seriously for the future. And there's lots of research opportunities, but there's also a threat to human health, and I will talk about this. So when you talk about antimicrobial resistance and antibiotics in general, quite often there's different messages. In the popular press, often there's articles about so-called superbugs, and I will talk what the superbugs actually is. And there's articles which are more or less trying to captivate the attention and the concern, the last resort. And this is an article, actually a commentary from Nature, which is linked to a paper published a few years ago, into which I will actually go into some detail what this was actually this paper, where there was a development of resistance, which actually meant to a lot of concerns a couple of years ago. But let's go look at the fact. And certainly when you look at the facts and the credible information, then certainly one of the best sources you can have is the World Health Organization, WHO, which considers antibiotic resistance as one of the biggest threats to global health, food security. As stated here, there's a copy from the website here, can affect anyone, occurs naturally, as I will explain, but misuse of antibiotics in humans and animals is accelerating the process. meaning that infections cannot be treated anymore, and this has important consequences. And I will elaborate further on this. Based on this realization, the WHO and applying governments from across the planet have implemented a global action plan on antimicrobial resistance in 2015, a couple of years ago. And one measure they implemented was a surveillance system called abbreviated GLASS. It's called the Global Antimicrobial Surveillance System. And GLASS, the goal of GLASS is, first of all, to observe what is happening. Because people talk about, when you talk about superbugs at the last resort, these are kind of alarmist announcement, but in the way you, of course, you want to look at the fact, what's happening. And GLASS is a system which has been implemented, is operating, and I figured it would be interesting for you to talk about, look at a country which you know much better than me, is Brazil, just to see what's happening, what are they doing, what is GLASS doing, what they're doing the monitoring. So GLASS, the first slide here shows a map of the world, of course, and shown in pink or magenta are those countries which have joined GLASS. So this global surveillance system. And you see, many, not every country, but most countries of the world have joined this system in a more or less intricate fashion and are joining and contributing to the global surveillance and data collection. Brazil, this I also took from the WHO website. Of course, big country, very high population as compared to Canada. Certainly, has joined GLASS. It's maybe not as deeply implemented in your hospital system, but as it could be in other countries. But it's working, and will be further integrated in the years to come. Just a few examples, and I'm not going to go into any details here, just to show you what data you can find, which is actually all from the WHO website. And this is actually from Brazil. So, for example, they look here at different bacteria and their resistance to antibiotic and the frequency here of the bacteria which live in the community, meaning they are not hospital linked. But then you also look at here blood isolates, which are actually isolated in hospitals. And, the data here in detail, of course, not important. But what I want to underline here is that, when you look, for example, here's, for example, one organism here, Klebsiella pneumonia. It's an organism which causes lung infections. Here 4% resistance you find in the hospital system. In the community, you find a certain lower level. But it's first of all, this is a monitoring system, and it's important, certainly, which helps us to understand what's happening across the planet. And of course, this is not always a peak like in a viral pandemic, but when these bars go more to the right and more to the right and more to the right over the next years, this will reflect the growing antimicrobial resistance or not, depending on what the developments are. But at least this global monitoring system, which will enable us to know what's happening across the planet. So, which is of course very important to managing antimicrobial resistance. The WHO has also published a list of very especially critical pathogens. This was from 2017. And here, again, the details are not important, and talk about, maybe some of you who are microbiologists know this, or have microbiology training, know this slide already, or at least speaks to you. It shows certain pathogens which are important as causing human diseases and their resistance here, for example, to carbapenem. And carbapenem is actually an antibiotics which are linked to belong to the family of penicillins, which are very frequently used antibiotics, but carbapenem are actually more advanced penicillins. They're not like the original penicillins, they are more advanced molecules, which are often used as something like a last resort antibiotics when others not working. And since some bacteria have developed resistance to these drugs, they are seen as critical, and we really need new antibiotics. And when you go down the list here, you see a lot of, I would say, usual suspects, bacteria which cause infections and which have developed resistance. And we'll talk about Staphylococcus aureus, which is a very important example here as well. Our own research is in Helicobacter pylori, and I will talk a little bit about this. Neisseria, causing sexually transmitted infections, but also brain infections. So this is a long list. An organism which is not on here, and I think the WHO was very criticized for at the time for this, is that Mycobacterium tuberculosis is and remains the most important bacterial pathogen across the planet. There's lots of strains, multi-resistance, and especially in Africa, this organism is important. It causes co-infection with HIV and causing no more than 1 million deaths per year. So 1.5 million deaths per year are caused by this bacterium, and the resistance is also on the rise for this organism. So it's certainly preoccupying in more developing countries, but also in developed countries, it's a distinction which is not always appropriate. But when you even look at Canada, seen as more like developed country, we also have Mycobacterium tuberculosis in certain populations. It is a concern for everybody, and not because so there's something to watch out for. So when you ask you what could happen in future if we don't do anything, if antimicrobial resistance develops in future. And of course, sometimes it's not easy to predict these development, but there's a lot of serious work has been done on this. And I would talk about two reports which came out. The first one attracted a lot of attention, the so-called ONeill Report, published in 2016, supported by the Wellcome Trust and the United Kingdom government. And this is a report which actually concluded that if things go as they proceed right now, there could be up to 10 million deaths per year in 2050 due to antimicrobial resistant infections in future, which I think is quite a significant, quite a high number. It's a very serious, I think, concern which has been raised. That how does it compare? Of course, numbers are often abstract. What does it mean? 10 million deaths per year. How does it compare to what happens now? When you look at here, for example, and this is also from the website of the ONeill Report. If you look at the slide here on the left-hand side, how many deaths happen now? In 2016, when this report was published, close to cancer 8 million deaths per year. This is worldwide numbers. Diabetes 1.5 million, diarrheal disease, traffic accidents 1 million. So traffic accident is something preventable, of course, but they happen, and it's part of our society. But right now, when you look at the antimicrobial resistance, it's about 700,000 deaths per year. Can be attributed to the phenomenon of antimicrobial resistance. And this number, which is much smaller than cancer, is not even a tenth, but it still is already a significant number. But if you don't do anything, if antimicrobial resistance continues to rise, if the tsunami continues to rise, this could go up to 10 million. It actually kill more people per year than cancer. Than cancer. The distribution is shown here on the right side, the predicted distribution, and maybe not surprisingly, usually developing countries are typically more affected by these kind of diseases than others. Maybe another comparison, right now everybody talks about COVID for good reasons, but up till now, and we are a bit more than a year in the COVID pandemic, COVID caused, at least according to official numbers, 5 million deaths, which is very significant. The unofficial numbers might be higher, could be maybe up to 10 million. Nobody knows really how many people really died of COVID. But in the end, if we don't do anything, antimicrobial resistance could cause the same or even more death per year in 2050. So this is certainly something to be taken seriously and not to be neglected. And this is why there's widespread concerns about this phenomenon. And another report was published by the Canadian Council of the Canadian Academies just 2 years ago. And when you look at Canada, our population, we are a smaller country, even if we live on a big surface. We have 38 million people in Canada as of 2020. And when you look at the infections in Canada, there are, about 1 million per year. So, just also significant people who have bacterial infections in Canada, and resistant infections, about 26% of that, which is already significant. So how many deaths does this cause? And, this is a study which was very complex and looked also at all kinds of factors, which I will not have time to mention. Of these 250,000 resistance infections, 14,000 people died. And 4 in 10, about 5,500 people this is lives which could have been prevented. So 5,000 right now. So, which is, it is a significant number, but that is the steady state. If we don't do anything, the the conclusion of this report was that, you know, right now 20% of infections are resistant to so-called first-line antibiotics, meaning typically when you when people are infected, have bacterial infections, are being treated by a doctor, there are certain antibiotics like penicillins and, you know, things which usually work. But even now, there's 26% of infections are resistance, meaning in this case, doctors have to find another antibiotic to treat the patient, which may or may not work. Usually, it's a more aggressive treatment, which may apply stay in a hospital, and intravenous application of the antibiotics, meaning it's much more challenging to treat. And, you know, some people die, because, you know, even if sometimes the so-called first-line antibiotics, which is often penicillin, doesn't work, then, you know, doctors may try another one, and then maybe another one. And it happens, unfortunately, that, you know, despite all the efforts, nothing works anymore. That's in more and more a situation we may be facing. The estimates are that, you know, if we don't do anything, and the tsunami keeps rising, this could increase to 40% by 2050. And then when you look at the effects this could have, we're just looking at Canada. Now, this could lead, meaning that in Canada, almost 400,000 lives could be lost over 30 years still, but it's a very significant number. Over the next 30 years, the number, you can calculate the cost this would cause in hospital expenses for a patient. Just for example, you have a patient who, you know, doesn't respond to first-line treatment, has to be hospitalized, may need a hospital stay. You can calculate the cost for all this. And this, you know, is very significant numbers. And of course, people who are in hospital, who cannot work, you know, this has different costs for society. So the estimates that up to $400 billion loss in gross domestic product could be caused just by the difference of 26% to 40% of antimicrobial resistance. You know, and then, you know, if you could reduce this, of course, this would be beneficial. But that's the situation as it is right now, and this is certainly has to be taken seriously. So now, I mean, people have talked a lot about, you know, what causes antimicrobial resistance, and people talk a lot about so-called superbugs. And, you know, here I found a slide online, you know, or kind of, you know, in the public imagination as different perceptions of what's actually be a superbug. And, you know, this is just like a very scary organism here, which, you know, can kind of fighting back all the treatments. But, you know, of course, that's not very scientific. But, you know, in the end, what are superbugs? And these are typically bacteria which have developed or acquired resistance to many or most commonly used antibiotics, meaning you cannot treat them with penicillin and many other antibiotics that are usually taken to treat infection if the first-line treatment doesn't work. Some of them are resistant to the so-called last resort antibiotics, and I will further elaborate on this term. A superbug is not necessarily more pathogenic. You know, here I just have to make sure that people really understand why the terminology is being used. A superbug essentially is mostly described a bacteria which is very resistant to antibiotics. The bacterium as such may not be more pathogenic, but the big danger is that since bacteria can very actively exchange genetic information, the superbugs may actually transfer their resistance to other bacteria, including pathogenic bacteria. If pathogenic bacteria then acquire resistance to very many different antibiotics, then we have a problem. And that's the process of bacterial DNA transfer, which I will talk about in more detail. So why is this now a problem? Certainly, antimicrobial resistance leads to increased mortality and mobility, and typically affects more vulnerable economically and socially more vulnerable populations. The consequence would be that so-called simple or benign infections could no longer be treated. And what does it mean simply? I mean, usually most of us don't have a concept that, you know, you're infected by a bacterium, that this would actually kill you. I think we don't think about this. I mean, we know viruses can kill us. There's very dangerous viruses, and there's right now, of course, COVID can kills people. But usually, bacterial infection, we think typically they can be treated. But, you know, this is unfortunately less and less true. And you know, I've showed before the list of the particularly preoccupying organisms by which cause certain infections, which are relatively common, which could be lethal in future if they can no longer be treated. Like, here it's a sexually transmitted infection, throat infections, lung infections, tuberculosis, of course. These are very concerning here. Medical procedures. Certainly one of the big effects of the introduction of antibiotics has been that it has changed medicine in a way, in a big way, since antibiotics can be used to prevent infections and to fight infection when they occur. For example, surgeries. I mean, surgeries like cesarean surgeries for the liver of babies, hip replacements, other surgeries of the these are surgeries usually we typically think these are of course primarily surgical interventions, but typically patients are being given antibiotic as prevention so that there's no infections after the surgery. If now antibiotics wouldn't work anymore, then a surgery, just a routine hip replacement, would become much more dangerous. And just imagine nowadays for people who are more senior, like hip replacements, knee replacements, it's a kind of a routine procedure once you have a certain age. And but this could be much more dangerous if antibiotics don't work anymore and cannot protect us. Cancer chemotherapy is another example. Is often antibiotics are given as prevention for cancer chemotherapy to support the patient if they don't work anymore. Also, cancer chemotherapy could be much more dangerous. And of course, we could face the re-emergence of bacterial pandemics. So are all bacteria dangerous? And that's something also a notion that sometimes, people, I mean, right now we're living in a way where everybody is wearing a mask. People are washing their hands frequently. And, these are kind of hygienic measures, which, people have, also applied to bacteria in the past. But, bacteria, I should underline that bacteria as such are not necessarily dangerous. I mean, in fact, bacteria live with us and in us. And that's quite normal. It is a fact that we are being colonized by bacteria. They live on our skin, on our body surface, on our hands. They live in our intestinal system in large concentrations. And in fact, what is called the so-called microbiome has many more cells than us. And people even talk about, this here now, again, like the cover page of a journal, Nature stating that bacteria are considered as the other genome, which has multiple interactions with us. And most of these bacteria are harmless commensal or even useful. They actually have important metabolic functions and help us protect against infection. There's even a movement, and this is actually quite interesting, a very interesting area of research, which has the aim to use bacteria to treat certain infections, meaning there's using mixes of bacteria as drugs, which is something which is still in the experimental stage, but for some diseases, and notably some gastrointestinal infections, actually the treatment with bacteria, here, as you see here, also from the Nature, the promise of poop. Essentially, you see the brown substance in the syringe here, which is actually that what you think, which is actually the fecal matter from a donor given to a patient who has a particularly difficult gastrointestinal infection. This is actually curative. This is actually something which is actually being used to treat certain infections, and bacteria have potential also for other treatment. But this is mostly in an experimental stage, but it's certainly very interesting for us. I'm just outlining this here to say bacteria as such are not dangerous. But of course, they even help us. Since they're colonizing us, they help us protect us from infections with other bacteria. But of course, there's bacterial pathogens which can become antimicrobial resistant, which are, the reason for this concept. Even most bacteria are not dangerous. So what do I talk about chicken and antibiotics? Just I want to give you. I realize there may be lots of people in the attendance of microbiology training or background. So this will not be news for you, but I figured this is a general audience. I spent two slides to talk about the development of antibiotics. I mean, this happened in the early 1900s, mostly in Germany. In Germany, for the Second World War, there were developments for the development of antibiotics. And at this point, since so little was known about bacteria, about the mechanisms, researchers look for something like the so-called magic bullets. They want to find bullets, weapons, could specifically hit and kill bacteria, but not the patients, of course. And the first magic bullets, this is actually a treatment used for the treatment of syphilis. Those of you who are chemically inclined may see here there's arsenic in here, meaning this is not necessarily a healthy substance, but it was the first treatment which worked against syphilis, which is a historically important sexually transmitted disease. Then in the 1920s, there was the discovery of penicillin. Certainly Alexander Fleming is certainly well-known name who actually discovered at some point, he noticed when he was doing his research, he actually worked on a bacterium which is called Staphylococcus, which is a bacterium which causes diseases, and especially wound infection. At some point, he noticed that there was a contaminated plate, agar plate, on which he colonized his Staphylococcus, and he saw that in the interface between this penicillin, fungus contamination, because he didn't want to study the fungus, and the bacterium, there was a zone. And he concluded from this observation that apparently the fungus, penicillium, produces a substance which makes sure that the bacterium cannot grow, because here the bacterium grows, here it grows, but at the interface it doesn't. So suggesting that the fungus produces something which stops bacterial growth, And this was a very important discovery, which was then further developed by other researchers in towards the Second World War. And this is actually the time when antibiotics were entered large-scale production and were actually produced, and especially during the Second World War, since there was a very difficult period in human history where there was lots of wounded soldiers, lots of people who needed treatment, because in a war, of course, people die of all kinds of causes, but a large proportion of soldiers or people who were wounded by gunshots or by other things didn't actually die of the gunshot, but they did die because with the gunshot or another wound, bacteria can come into your body, infect you, and this could kill. So the Second World War was a major motivation for the development of antibiotics, but of course there's many different needs for this development. Then, there was a period where antibiotics were discovered in many environmental organisms, especially fungi and bacteria. And quite often this shows you here a plate where there's actually not a fungus, it's actually a bacterium growing on a solid medium. And what you see in blue is actually substance which has antibiotic activity. And, not all antibiotics are colored, but some of them are. And this is just an example here. So there was lots of discovery was done, and thanks to this discovery, formerly incurable diseases were suddenly curable. And this slide here shows you the structure of penicillin. Some of you may be more chemically inclined. It's not very complex substance, which was one of the first antibiotics, and this became immediately very popular. And you see here actually a letterbox. This is, I think, from 1930s and 40s. You see penicillin cures gonorrhea in 4 hours. A sexually transmitted disease which could be cured now can be treated. And so this is actually publicity, here on a letterbox, in the middle of the last century, because antibiotics were seen as wonder drugs, because before antibiotics, in the pre-antibiotics area, people died of infections, something we don't realize, because we're not used to people dying from bacterial infection, but this was important in the past, and all of a sudden they could be cured. So this was quite an important development. Changed me, I think. And how do we come with chicken? Why do I want to talk about chicken? Here's actually a book I find very interesting but I'm not here to promote the sale of this book. But I just want to give you some information which I found very interesting in this book, and which actually make a link to agriculture. Something which not everybody, probably not many people in the audience may know here, is that antibiotics were, of course, developed for treatment of infections in humans. But in the 1950s, discoveries were made that when you use them in agriculture as feed additives to livestock, this actually helps the chicken grow faster. And the same works for cows and other animals which are livestock, which are usually being eaten. And the mechanism why this actually works is not entirely clear. So, I would like to underline, I'm not talking here about antibiotics used to treat diseases in farm animals. No, this is really, it was discovered that, if you add a little bit of antibiotics, they grow better. And this of course helps with the output of the agriculture. It was widely adapted, and antibiotics were also used for food preservation. So what happened actually? So the chicken were cultivated and reserved and received antibiotic in their feed, but they were also after the treatment after the chicken were at some point slaughtered, and then they were dumped into a solution of antibiotics to preserve, to make them sure that they can be preserved longer. And this was, practice in the 50s. Unfortunately, it was, and the thing is that even now, when you look at the numbers, the tons of antibiotics which are being used for human medicine and compare this to use in agriculture, is actually more antibiotics are being used for agriculture and not to treat diseases. Well, treating disease in animals is one thing, but actually most of the antibiotics are being added as feed additives because they help the animals grow faster. So, which still works, but of course, there's unfortunately consequences. This was also discovered in the end of the 1950s that there were in operations which were linked to the maintenance and to the treatment of animals after slaughtering, there were disease outbreaks, and this was certainly a warning sign that people who worked in these facilities developed infections that could not be treated, which again signaled it was a really a red flag that apparently resistance against these antibiotics had evolved, and these disease outbreaks led to consequences for the use of antibiotics in agriculture. So when did AMR appear? Actually very soon. And this slide here from textbook, the Brock shows you, and again, the numbers here, the names of the bacteria are not so important, but I'm showing this here. If you remember correctly, antibiotics were developed in the 1930s, 40s of the last century. When did resistance appear? Well, actually pretty soon. Staphylococcus aureus, which is a particularly preoccupying pathogen, started researchers and doctors and medical doctors started noticing resistance beginning in the 1950s, which is relatively fast after the introduction of antibiotics. And now when you look at the bacteria, it's like, a list of laundry list of important bacterial pathogens which developed resistance successively over the last 50 years. Give here some more information. The details are not so important. Staphylococcus aureus did not develop only resistance against one antibiotic. This slide actually I think is quite telling, showing here's Staphylococcus aureus developing resistance, and when you see a Staphylococcus aureus organism in the 1930s here, before the introduction of antibiotics, was sensitive. S stands for sensitive, of course, to all these antibiotics. These are really all kinds of antibiotics you could think of all these different classes. But here at the end, towards the end of the last century, Staphylococcus aureus had developed resistance against most of them. And even against so-called, what is called reserve antibiotics. There are some antibiotics which are usually used as a last resort. Meaning usually when people have an infection, the doctor uses so-called first-line antibiotics to treat, which usually work. If they don't work, you try something else. If they don't work, try something else. But sometimes it's like, you have a patient who is really in trouble, antibiotics don't work anymore, you give a last resort antibiotics, the reserve antibiotics, like vancomycin. But Staphylococcus aureus has also developed resistance against this antibiotic at the end of the last century, leading to bacteria which can essentially not be treated anymore. There's very few options which are available. So this is really certainly quite impressive, the ability of the bacteria to adapt to this, to all these antibiotics. In parallel, something happened which may not be easy to understand, but I think in retrospect could be understood. There was a so-called innovation gap. And this slide here from 2009 shows that there was a lot of development of antibiotics in the 1940s, 50s, until the 70s. Lots of new class, there was lots of discovery, lots of research going on, lots of discoveries of novel antibiotics. And then something happened. No major classes of antibiotics were being introduced. And this is actually leads what some people call a perfect storm. When you see here, and this slide here on the left shows that the kind of a curve which goes down, meaning the companies which actually are doing research on antibiotics going down. Companies apparently lost interest, and which considerably slowed down the development of antibiotics. There's right now very few companies which actually work still on the development of antibiotics. At the same time, resistance is increasing. And these are here just some examples. I don't go into details here. Resistance goes increasing, and companies who work in antibiotics going down and down and down. So why, and why is this happening? So you could think, why is this happening? And didn't we win the fight against infectious diseases? And this is actually maybe part of the answer, because we thought with all the antibiotics developed here early in the last century, that we had actually won. And usually, I mean, to be serious, often there were situations where there's a first antibiotic works, and it still works in many cases. But the numbers I showed you before in Canada, and probably the numbers are pretty similar in Brazil, 26% the first line of infections, the first line antibiotics doesn't work anymore. So you need a second line antibiotics. But still, there was a notion, at least at this time here, that we actually did win. So this kind of slowed down the interest and also the economic interest, because at some point, of course, there were so many drugs available, and developing a new drug is very costly, costs up to hundreds of millions of dollars usually to develop new antibiotics. So for companies who actually want to sell antibiotics, the economic incentives were not so important anymore, leading again to this decrease in the interest for the development of antibiotics. This has changed. But we are certainly still playing catch up. Certainly in the beginning of the 2000s, there was some development, but still there's not a lot of really, when you think about what companies are working on, they work on cancer drugs, they work on drugs for the aging population, now on COVID drugs, but antibiotics, that's not a big market for companies, which also has an economic, so there's also economic questions, and I will touch on this briefly later on. Now the question is, so where do we find antimicrobial resistance? Something you may ask yourself, where is this actually happening? Is this something only in hospitals? And here this actually shows, the hospital which is affiliated with the University of Montreal, a so-called super hospital with the highest standards of care and the research center right next to it. And yes, of course, you find antibiotics in hospitals, but you also find them in the community. And here you ask the question, where does it come from? Is it something which evolves in a hospital, or is it from somewhere else? Questions which are very valid, you can ask yourself. And in fact, when you do research, and here I'm talking about some publications that came out over the last 10 years, which ask the question, do antimicrobial resistance? Of course, you find them here in hospitals and patients. But what if you looked now in nature, in wildlife, in areas where, there's not many humans, there's no hospitals, there may not even fields where there's feedstock. But when you do sampling, this is actually a publication which came out in 2006, which was done by a lab in McMaster University in Canada. In Canada, where researchers actually went out into nature and went to really, really far away areas, conducted soil samples from all kinds of areas, but typically far away from human influence. And what they found resistance against all these antibiotics. They found that bacteria isolated in these areas were often multi-resistant, meaning being resistant to up to 10 different antibiotics, and also some of the very new ones. And I'm not talking about the details here, but some of these bacteria which come from really different environment, which are far away from human influence, there's antibiotic resistance is there. So it's in many ways a natural process, can be found everywhere. This led to the development of a concept of the so-called resistome, implying there's lots of resistance of bacteria happening, and it's the genes are somewhere out there in nature, play a biological role, and it's mobile. And that actually brings to the next topic, that the fact that antibiotic resistance is maybe somewhere in nature in different environment is not necessarily a problem if it stays there. But, of course, since it can be transmitted, and this is something I will go into more detail, that's actually can lead to concerns, since these antibiotics can come closer to where we live and even to hospitals. Another question you can ask yourself, is antimicrobial resistance recent or old? Is it something only, antibiotics were developed in the 1950s. last century, 70 years ago. What about 10,000 years ago? And this is actually a publication from 2011, where researchers actually went to a site which is far away from human influence here in Yukon, in the north of Canada. And they actually found a site which actually had been frozen for 30,000 years. 30,000 years, so a long time ago. So they took samples from soil which had been frozen for 30,000 years, so way before the development of antibiotics. They found antimicrobial resistance genes against many currently used drugs. And here, some of you may be inclined to do more biochemical work. They actually showed here, they even actually cloned the genes and expressed an enzyme coding for antimicrobial resistance, and showed here, when they compared the structure, the X-ray crystallography structure of the ancient enzyme, 30,000 years old, and the new one, and compared the structures, they found this was actually very similar. So, 30,000 years old, this is an old process, it's a very old process, so it's nothing recent. So I think this is quite stunning to see, but, here we talk about, what you find in nature, and which helps us also understand why this happens, how it's being transmitted, and helps us also devise strategies to do something about it. The next slides, I will talk more about mechanisms. How does antimicrobial resistance actually work? I mean, how can bacteria actually become resistant to antibiotics? And they have different mechanisms, and I will talk about some of them. One of them is permeability, meaning antibiotics cannot come in, but bacteria also have the capacity to pump antibiotics out, which is especially difficult, and I would say annoying process from the human perspective, since antibiotics can be pumped out of the cells. Antibiotics can also modify and destroy the antibiotics, or actually protect the target in the cells. And the next slide, this is more for the biochemically inclined. I'll show you I go a little bit more into details here. And this structure here shows you on the left-hand side, shows you the envelope of a bacterial cell with something which is called an antimicrobial resistant pump. And it's a cryo-EM structure, some of you may recognize it, showing a pump is actually a macromolecular structure comprised of proteins, which has the capacity to take antibiotics which are inside the bacterial cell and flush them out. Meaning the antibiotics goes in, may find a target inside the cell, could in principle kill the bacterial cell, but the pump flushes out, meaning cannot impact the cell anymore. Bacteria also have the capacity to actually protect themselves by modifying the internal structure. And one target, which is especially well known, is in the ribosome of bacteria, the ribosomal RNA, and the biochemist between you will certainly recognize the structure here, which is it's a conserved loop in the ribosomal RNA of bacteria where some antibiotics bind, like erythromycin. But some bacteria have the capacity to actually modify this ribosomal RNA and are not vulnerable anymore. So this is the way they can defect it. Probably the most well-known mechanism for bacteria they use is they can modify, destroy the antibiotics. Essentially here showing you penicillin, the structure of penicillin. The bacteria actually have a mechanism to go and chew up this molecule, and they kind of have a way to cleave here. And when you have here penicillin, you cleave this chemical link here, penicillin will not work anymore. So these are kind of mechanisms bacteria use to destroy antibiotics, and these mechanisms are mobile, and that's actually part of this slide here may remind you of what we are talking about right now a lot in the case of COVID, because COVID travels with patients with humans, and antimicrobial resistance travels in the same way. And this is actually a slide taken from an article I've alluded to before, when there was a certain resistance development was preoccupying, which actually was first discovered in India, and due to global travel, then traveled across the globe and came to different countries. And this is actually the article here where they actually taught this so-called last resort article, talking a bit more about the science. First of all, this antibiotic was discovered in India. First, then transmitted and discovered in different countries, North America, And since this antibiotic causes resistance against, I will not go into detail here, causes resistance in bacteria, which already carry many other resistance organisms, it was concerned. Why was this particularly a concern? Why were this headlines, the last resort, why was this so preoccupying? It was so preoccupying because these bacteria, these so-called NDMN1 producing bacteria, is an enzyme which inactivates an antibiotic, degrade this antibiotic. And this is actually, those of you who are chemically inclined may think, oh, this looks a bit like penicillin, right? But it's not penicillin. It's a very evolved penicillin. It's kind of a 5th generation penicillin, which has been developed over many years and is a so-called reserve antibiotic, meaning an antibiotic which is used when nothing else helps. Patients which have been treated with first antibiotics and they're really in trouble. So when the doctor has the last reserve, we use this antibiotics. And here there were bacteria which were resistant against even this antibiotic. This was really very preoccupying and led to these articles, which I alluded to before. How, the spread of antibiotic resistance shown here, but how do they do it? How can bacteria spread antimicrobial resistance that efficiently, especially how can this come to pathogens? And here, I think I have 2 or 3 or 4 slides left here of this part showing that antimicrobial resistance, bacteria can do different things. They can mutate the genome. They can actually take up DNA, eat DNA, but also incorporate it into their genome. But the most frequently used process is so-called plasmid conjugation, meaning bacteria have the capacity to take pieces of DNA and transfer them between bacteria. And this actually, I have a few slides about this. Is also actually close to my own research, which is linked to the fact that bacteria have a genome, a chromosome, and plasmids, which are kind of extra chromosomal elements. They are small DNA snippets, which are not in the main genome, and they can be transferred. So bacteria have the capacity, using a mechanism of translocation, to translocate plasmids from one bacterium to the other. And imagine this is a so-called superbug, which is maybe not necessarily pathogenic. But if then this superbug transfers this plasmid and the antimicrobial resistance to pathogenic organisms, then we have a problem. It's a process we don't, even if it was discovered 70 years ago, we still don't really know how this works. And this is a very suggestive picture, from 1971, which is like, I was a kid in 1971, and I was not a kid yesterday. This is a long time ago. And this is actually an image of one bacterium here, which donates DNA to another bacterium, while the structure here, which links these 2 bacteria, and this is a process which probably involves the cells fusing, which, again, this shows you here again, 1991 is not very recent. It's a process which we understand not completely, but we understand a bit more. I mean, the process which is used, and this is probably only for the interesting for the biochemists in the audience, there's a so-called type 4 secretion system, a complex in this bacterial cell envelope, which is actually used by the bacteria to translocate. And I will show the structures, but I don't really think we need to go into detail. It's a macromolecular complex in the cell structure of the bacteria, which actually helps the bacteria to translocate from the inside of the bacterium to the outside into other cells. And we are using approaches of structural biology to more and more understand this process. Here's a much more recent work using a so-called technique, which is very advanced, which we use a lot here in Montreal, at University of Montreal and at McGill University in collaboration, showing images of bacteria with these structures at the surface, which the bacteria use to actually contact and touch other bacteria to translocate DNA. We get kind of trying to getting a picture how does this actually work, how does this look, and there's an approach which is called so-called tomography, which actually helps us more and more to look inside bacterial cells. Bacteria are very small, 1 micrometers, and then we can actually zoom in with microscope directly to see the structures which are doing. We can also look at the surface structure. This is actually a picture from my own lab, which is actually just a few weeks old. So we actually look these so-called structures here on the surface of the bacteria, which they use to touch other bacteria. We can use them by electron microscopy. You can stain them, but we also use cryo-electron microscopy. Actually, we solved the actual structure of one of these surface structure, a so-called pillows, a while ago. And you see here, there's a hollow tube with a hole in the middle, which probably serves to translocate DNA And it was also discovered, which is actually quite fascinating, that is a complex made from a proteins and so-called lipids in the. But I think this is something, if there's more interest, we can discuss this further. It's probably only of interest for the biochemically inclined here, but this is certainly very recent research in my own lab, and other colleagues in North America and in Europe work on this as well. So what can we do now? And closing this part of the talk, certainly what can be done about antibiotics is clear. We need to develop novel antibiotics and combination therapies. Certainly a question which is not linked to usually the natural science and the medicine, but as I mentioned, for many companies it's not really interesting to develop novel antibiotics because there's no economic incentives. But this is also something which is actively being worked on by different measures taken by governments to actually make sure, and this is really something evolving, to make sure that there should be more interest in companies to develop antibiotics. It's certainly something which would be very helpful to change the situation. Needless to say that existing antibiotics should be used responsibly, only when they are needed. And certainly the question is the use of antibiotics as growth promoters in agriculture is certainly something which is certainly preoccupying, and certainly should be limited, and has been limited in many countries already. What we also should be doing, of course, improve diagnostics and monitoring, and the GLASS system I introduced before, so that we see what's actually happening. Surveillance, we need to know what's happening, what the tsunami is coming, which will allow us to predict and develop novel approaches. And last but not least, also an area I'm myself very interested in, is the development of alternatives to antibiotics, like vaccines, phage therapies, antivirulence drugs, and inhibitors of the conjugation process. So there's lots of strategies out there to do something, but there's really ongoing research. And I'm mentioning this because it's important certainly to show what's been happening and to develop strategies to counter this development. So just to make sure that this tsunami which is rising is not flooding us and washing up away. But it would certainly be require coordinated efforts across the planet to do that. I'd like now to finish just with a few examples of research opportunities at University de Montreal, show you a few researchers who have done very interesting research, like Dr. Brun, who was recruited 2 years ago. He works on bacterial cell biology. He's really a basic researcher. And what he wants to understand is different bacteria. And he really looks at environmental bacteria and really wants to understand how they operate, how they function. For example, he found bacteria which live in water and which actually hold on to surfaces with a glue. And this glue is actually the strongest natural glue, stronger than than industrial, or as strong as industrial glue. So this bacteria can stick to surfaces. He has shown he is using fascinating approaches of imaging, of microscopy, to show these surface structures which the bacteria use to move across surface. They can actually dance. Some people call it dancing pillar, and this is actually an image here. It's a movie of a so-called dancing pillars. This actually shows a bacterium having one of these pillar, which actually sticking, shown in red here, pieces of DNA. So these are pili which they're dancing around. They can actually take DNA, take it up, and this DNA this bacterium takes up could actually contain an antibiotic resistance gene, which is part of the process which leads to the spread of antimicrobial resistance. Bacteria can exchange and take it up, and what he certainly wants to do, he's a basic researcher, but what he's doing is extremely important since he wants to find novel targets for antibiotics. Dr. Daigle is also in the microbiology department. She works on Salmonella species, which is an organism which is very dangerous, especially in developing countries, killing lots of people, especially children, causing food poisoning. And she wants to understand how this organism evolves, wants to understand the molecular basis of the operation of this organism, and especially want to understand how this organism adapts to different species, to humans and animals. So there's also researchers who has very interesting projects. Diana Falcone is actually working at the University de Montreal affiliated research institute, and she's actually an MD, is a doctor. She's interested in the microbiome. So she doesn't really work on pathogens, but she wants to understand how the microbiome, the organisms which work in us and with us, actually cause certain diseases. And diseases she's interested in are mostly intestinal diseases, which are hereditary, which cause for the patients who are affected lots of problem, constant inflammation, constant diarrhea, and she wants to understand how these genetic diseases cause this process, which is actually quite frequent in Canada. And she's an MD, she's a medical doctor, and also works in long COVID syndrome, and want to understand how the microbiome and the immune response interact, and to find better treatments for these inflammatory conditions. For some reasons in Nordic countries like Canada, also Northern Europe, these diseases are more frequent than in southern countries and warm countries like Brazil. So there's a significant part of the population affected by these diseases, which are linked to the microbiome, and she wants to help understand how this works and develop better treatments. Dr. Finzi is one of our superstars. He works actually, I showed the hospital I showed you before. He works actually in the research center of this hospital, and he's actually a specialist on HIV. So he's a virologist, and like many researchers, and also Dr. Finzi, last year, for reasons we all understand, he actually switched part of his research to work on SARS-CoV infections, and he's actually somebody who does very interesting work looking highest resolution electron microscopy approaches to look at how the virus binds to the cell, enters the cell, and he also studies the immune system. And he wants to develop innovative approaches to understand the immune system response and to block entry into the cell. Also, Bertrand Routy is a colleague of him. He's a medical doctor as well. He's also in the area of microbiome research, and he's interested in also a very topic I find very fascinating. He wants to understand how bacteria and cancer interact. I mean, there's an approach which is very promising, has been seen as increasingly promising, as it's called the cancer immunotherapy. It's a therapeutic approach to treat cancer. And he is one of the pioneers who actually found effects on the microbiome, meaning the bacteria, which are usually harmless, don't cause diseases, but there's some bacteria. If you have some bacteria, cancer immunotherapy works for you. If you don't have the right bacteria, it doesn't work. And who are the good bacteria, who are the bad bacteria, and why some people don't have the good bacteria and therefore cannot be treated with cancer immunotherapy. That's something he wants to understand. It's also very fascinating research, again, it's not really linked to antimicrobial resistance, but he wants to understand how does the bacteria which live in us, how do they contribute to our health, and how can they be exploited to advance treatment. Dr. Serojicos, a colleague of mine, he's an evolutionary biologist. He's a more computational, he has lab-based research, but he's also a computational researcher. He's somebody, his research in the area is the evolution of antimicrobial resistance. He wants to, he wants to understand how does these things evolve, wants to look from what we know now, wants to look into the future, and hoping to develop and to actually predict antimicrobial resistance in the future. And this is something which is also very, very interesting research. He's also interested in COVID research, because like many of us, we would like to see how the pandemic is working in future, but also certainly working on this slow pandemic of antimicrobial resistance is also important to use all our imagination to predict of what's happening. And just last but not least, I talk a little bit about my research lab with the last slide. We are interested in the process of conjugation, and I showed you a slide before, but we also work on a bacterium which is called Helicobacter pylori. It's actually a bacterium which is very widespread, causes in about half of the human population. Pathogenic strains of Helicobacter cause actually stomach ulcers and even stomach cancer, and this is a bacterium which lives in our stomach, has a very interesting curved shape, has surface structures which actually the bacterium uses to touch human cells, and then to inject toxins into human cells. So this is always we try to understand, and our research is also very linked to. I'm a microbiologist by training, but we do a lot of biochemistry and also structural biology. Since you want to develop small molecules, in this actual structure for my lab, where we actually have an enzyme which is part of this machinery, which erects the structure on the surface of Agrobacter, of Helicobacter pylori, and we actually found a small molecule which inhibits this machinery, and which we will hopefully will apply in future, and hopefully will be able to develop better treatments for people affected by this organisms. Yeah, to conclude in thanking, my co-workers, there's currently 6 researchers, PhD students, master students, postdoc, undergrad students in my lab, and our collaborators and the funding system. So certainly, I'm talking here for them and representing them, but the work in the lab is done by researchers, people like you. Many of you are in the audience, I think, who want to conduct research in future, and I hope that this presentation has given you an overview on the research which is being done at University of Montreal in this particular field, and you will hopefully participate in other presentations in the context of the tech journey, where other fields of research are being mentioned. And I would like to, with that, thank you, merci, danke, gracias, obrigado, and would like to thank you for your attention and your interest. I have a few more slides actually to show you, and this is the end of my presentation. I think before we go open the session of the questions, I think I show the other slides we have, because I think lots of you have questions here on research on our institution, on Université de Montreal. I think I have 5 slides here, and we are very pleased today. We have Carla in the audience, who is actually a specialist from the recruitment office, who is there to answer to all your questions on our institution. Certainly the scientific question on antimicrobial resistance research I can answer, but we have Carla here, who is probably much better than me. This one we don't use. Let's see, I have the right presentation. Let's close this one. So just a few more words about our institution. Oh, can present this. Yep, here we go. All right. Get there. Okay, so why would you want to study in Montreal? I mean, and how? And this is something, here a few slides. I mean, our institution is actually the largest institution in Quebec, in our province. It's the second largest institution in Canada. We have almost 70,000 students, thousands of professors, and almost 5,400,000 graduates around the world. So we are training a lot of students at all levels. When you look at the research activities, we are number 4 in Canada, so it's one of the top universities in Canada, also highly ranked in the international comparison, like the Times Higher Education ranking. What are we studying? I mean, I think I shouldn't explain what you can study at UDM. I think there's few things you cannot study. I mean, we are really a big institution. We cover all fields of research: management, life sciences, medicine, the humanities, engineering, cultural studies. So, I mean, in the end, when you look at the programs, and it's not my role here to explain all our programs, you have hundreds of programs in all fields. I think it's difficult to imagine a field of research you cannot study at our institution. So really have a wide variety of offering, in all fields of research. When you think about Montreal, of course, studying is important. You want to have quality education. You want to have competent teachers, interesting colleagues, but you also want to have fun, right? Especially when you're young and when you're a student. And certainly Montreal is a place which is very popular because you can work here. The quality of the education is of the highest international standard, but it's also a very good city for students. And when you look at our population, Montreal has about 3 million inhabitants. If you look at the larger Montreal area, and we have about 200,000 students. So there's almost 10% of the population is students, meaning it's a very young city, very cosmopolitan. People are from all kinds of places. And most of our students are bi or trilingual. So I mean, here I speak in English, but as you probably know, Université de Montreal is a French institution, but usually students speak English and French. And when they're immigrants, like me, we usually speak a third language. So I speak German at home. And many people in Montreal actually speak two, or at least two, or not, if not even three languages. And this makes a very cosmopolitan city and very lively and very attractive, especially for young people. I mean, there's guides. I'm sure there's information you can find on our website. And Carla is here to ask all questions. But there's lots of information you can find. Also in English language. I mean, we're a Francophone institution, but certainly at the graduate level, we have a lot of students who are not Francophones. So we train, of course, we always encourage. I mean, French is, the French culture is so rich and interesting. We encourage everybody who comes here to learn and immerse in French, but you can have your training to a large part in English and hopefully we learn French over time. Really, we're really very open and accommodating for everybody who wants to come here. And I think that's the end of this presentation. And I'm sure Carla, and I think at this point we can go to question period. And I'm sure Carla will be happy to answer any questions linked to studies to which I cannot answer. With that, I thank you for your attention, for your time, and we'll be happy to discuss further. Thank you.
Hi, Carla. Welcome. Thank you so much. Professor, thank you so much for the talk. It was really interesting for all of those of us who are not in the microbiology segment, and we hear so much about it. We have some questions that people sent to us, and I'm going to read you the first. It's from Ronaldo Ramos, who is the president of CCBC. Dr. Barron, I always thought that every living being had at least one powerful predator on Earth, but it does not seem to be the case with superbugs. Is this correct? Apologies for my very basic question, but my background is in engineering.
I mean, bacteria have a limited lifespan, and they can in the end there's always a balance. I mean, a bacteria can grow. And like us, we grow, we live for a while, and then we die. And even superbugs, at some point they may not be able to resist, and they may not be able to be killed with antibiotics. But at some point, also they find a natural end. So it's not like superbugs are flooding the planet, are flooding the world. At some point they also reach their limits. And for example, a bacterium which is very pathogenic and kills a host cannot move anymore. So, it could be the end of the superbug. Imagine you have a superbug which kills me, I'm dead. And then, that's also the end of the superbug because it may not be able to spread anymore. So there's also a natural limit, and, nature, nothing dominates nature. There's always a natural end, and nature finds its balance eventually.
We have another question from Elena Leal. Excellent lecture, Dr. Barron. I'm amazed. I would like to know your opinion on ways we can handle resistance bacteria in wastewater. Are there methods focusing on biochemical aspects of microorganisms that can be explored as targets for water treatment? Thanks in advance.
Mm-hmm. I mean, in the end, certainly the monitoring of wastewater is an important approach. We can see what is going on, but in the end, the wastewater is not. In the end, I think if we want to prevent that bacteria with antimicrobial resistance come to wastewater, we have to start earlier. For example, use antibiotics responsibly, not flush them down the drain, not use them excessively in agriculture. This will also limit the number of bacteria which are antimicrobial resistant in wastewater, because once they're in wastewater, they are out there and can essentially transmit the antibiotics resistance. I think with antibiotics we have to start earlier, but the monitoring of wastewater is certainly very important. It's also done for COVID research right now. As you know, probably Brazil does the same thing. We monitor with the wastewater to see how many infections, because infections are somewhere else, but in wastewater we see it. We can monitor what's happening. So wastewater is very important here.
Just another, this is my question is, I wrote it as you were speaking, Professor, that it really amazed me that, I was Googling it as you were speaking, that in 30 years, the Earth population is expected to grow by 40%, but death caused by resistant bacteria is expected to grow 1,300%. It's a much larger growth than our population. Does this mean that bacteria are mutating and changing faster that we can respond to it, or that if the efforts that we are doing to contain infectious diseases are causing those bacteria to evolve faster, and that we are losing this race against these mutations and changes?
I'm actually more optimistic, and I think, but we have to get our act together and start entering the race. I think we have seen last year a virus, not a bacterium, which was kind of spreading and causing all kinds of things. And within a year, we have beaten it. I mean, there's lots of issues to still be solved. And as you know, when you talk about COVID, the fact that some people for various reasons don't want to take the medicine or the vaccine is an issue. But from a scientific point of view, COVID is done, is over. There's vaccines, there's treatments coming on the market. We have beaten it in a year. Antimicrobial resistance is more complicated because it's more diverse. It's not one virus, one target. But I think if humanity puts its imagination to it, we're going to prevail. But we have to be vigilant. We have to monitor and do what is necessary to be aware of the tsunami which is coming and react and prepare ourselves. But I'm optimistic that science will prevail.
So another question, we are causing bacteria to become stronger. And we do this through various practices, including some which are taught at medical schools. That the way we handle, doctors handle antibiotics and the practices that we have. Are those practices being changed in our medical courses?
I'm pretty sure that nowadays a medical school is at a very different level, and what is being taught on the use of antibiotics. When I was younger, and this is like stories from my childhood. My dad would come home and show an antibiotic prescribed by the doctor against the flu. Nowadays, this is almost a case of malpractice. I mean, nowadays, no doctor trained nowadays would prescribe antibiotics against the flu because it doesn't work, right? Flu is caused by a virus. But 30 years ago, maybe there was a different perception, but nowadays the training in medical schools is certainly up to date, and doctors are aware of what they have to do and when they can prescribe antibiotics and at what point, and that this should not be abused.
Okay, Professor, just one last question. This is an interesting one. Due to the pandemics, we all learned a lot about vaccines against viruses. And, there are lots of different types of vaccines and, lots of new technologies that came on, and now no one, most of, regular people didn't hear about, like vaccines that work on a genetic level and things like that. Are there any breakthroughs going on and on the development of antibiotics?
I mean, research at the research level, there's new developments, new molecules are being developed. What holds us back is that is not certainly, I mean, the amount of research is being done and being financed. I think we need to go much more broadly and develop many more molecules, because antimicrobial resistance is not caused by only one organism. And cannot be beaten with one vaccine or one drug. I mean, COVID in a way is an easy target. It's one virus. It's mutating, but it's one virus. It has about 10 targets, proteins. But bacteria, if you think about difficult bacteria, there's about 20 to 30 out there, and others may evolve. So to beat those, we need much more research, much broader operation to develop new molecules. They have their breakthroughs, but then when you have a breakthrough, the question is what comes to the clinic and who is going to develop it. And here I come to the question I alluded to, which are the economic incentives. Imagine you're a pharmaceutical company. You spend $500 million to develop the best of the newest antibiotic, which is the so-called reserve antibiotics. And then usually what doctors are being told and taught, and that's correct, don't use a reserve antibiotic, because the reserve antibiotic, the newest, should only be used if nothing else helps. So what do you do as a company now? $500 million, you want to sell your antibiotic, and the first message is don't use it. From the medical point of view, it's okay. From the patient point of view, it's okay. But for a company who wants to, who's give the company $500 million, right? I mean, we need to look at economic models, and there's different things are being discussed, which are also, we have seen examples here in the case of COVID, where governments made some investments, and to make sure that the research is being done, which may fail. I mean, as those of you who follow COVID research know, lots of vaccines were developed. A few of them work. Those who work were extremely well, but many more were developed. And there was lots of money was pumped in because there's such a big problem. But we have, my feeling is, if we do the same thing, or maybe not use similar broad strategies, which include the economic incentives for companies who are usually the ones who can develop new medications, we can also proceed more with antibiotics, because there are certain things a research lab like mine can do. But I cannot, the researchers do certain things, but at some point companies have to take it up and bring it to the next level and ultimately to the patient.
So thanks, Professor. I would just call then Carla from the University of Montreal to say just the last few words, but thank you ever so much for the talk. It was really enlightening. Really loved it. And it has been really a big pleasure. Thank you so much, Professor.
Thank you.
Oh, Carla.
Thank you very much. I think we all learned a lot today. Obrigada a todos por terem participado. Se vocês tiverem alguma dúvida sobre a Universidade de Montreal, a gente trabalha, então, desculpa que eu estou ouvindo a tradução ao mesmo tempo, vocês podem escrever para a gente no nosso Instagram, o DM Brasil, também temos um e-mail, brasil@oumontreal.ca, então a gente responde em português, se vocês quiserem se candidatar para algum mestrado, algum doutorado na Universidade de Montreal, saiba que tem uma comunidade brasileira na UDM. Muito obrigada a todos.
Pessoal, boa noite a todos, muito obrigado.